Today we are going to learn about apomixis. Apo meaning without, mixing of gametes. So if no mixing of gametes, no fertilization, no zygote formation. But over here, without fertilization of gametes, that embryo formation takes place. So how does this possible over here? That we are going to learn about that. Apomixis, first of all, it's a special mechanism. For that, certain apomictic genes are present. These genes are not present in each and every plant. These genes are present in some of the species of Asteraceae and grasses. Here the seeds are formed without fertilization. So now when we see seeds, the seeds basically form during sexual reproduction. It's not a part of asexual. So apomixis mimics the sexual reproduction because it forms the seeds. But here no gametes are required, no gametes are formed. So gametes are not formed. That is the part of asexual. So apomixis, basically we consider it's a form of asexual kind of reproduction, but it mimics the character of sexual reproduction. Which character? It forms seed, so sexual reproduction. Asexual meaning without gametes. Now in this apomixis, generally in the normal case, that haploid egg cell is formed. But in apomixis, diploid egg cell is formed. This diploid egg cell is formed from the which one over here? That's the nucellus is there. We have learned nucellus, one of the cell that acts as a megaspore mother cell, which is diploid. Here this cell has this apomictic genes. And now as and when these genes get activated, this diploid megaspore mother cell, first it skips the meiosis to form that megaspores and then the female gamete. Here this megaspores mother cell that will undergo directly mitosis 3 times and form that normal embryo set in that 7 cell, the 8 nucleate condition. But haploid condition will not arise due to the absence of meiosis. So diploid egg cell is formed, and diploid egg cell due to that apomictic genes converts to by itself diploid embryo. So apomixis, 2 main keywords remember: no meiosis, no fertilization, even third one, no gametes here. So diploid egg cell is there, gametes fusion is not there. So diploid egg cell that converts to directly diploid embryo. This we have to remember for the apomixis. In this, one last thing extra which you should remember for the meat. In case of diploid, that's apomixis, embryo sac, ploidy if asked, then it's a diploid. Because diploid cell undergoing mitosis, so it forms each cell in diploid condition. Normal condition, if there, we consider the female gametophyte or embryo sac is haploid. But now this one we consider as a diploid, that ploidy will be changed. Next that we are going to learn about polyembryony. Poly meaning many. Many embryos in each ovule or seed. Examples, again not seen in each and every plant. Examples here, that's the citrus and mango. Very, very important line for meat. Now, again, this is the structure of ovule. Here the nucellus is there, and inside the nucellus, normal embryo sac formation is there. Here gametes form, fertilization occur, everything is normal. But polyembryony, extra embryos are formed, then we consider many embryos are there. In case of nucellar cell, if we can make out this blue color cells are nucellus, this nucellar cell, some of the nucellar cells, it starts dividing mitotically and form a ball of that cells we call here the embryo, and that embryo pushing towards this embryo sac. Why pushing towards that embryo sac? Because we know earlier we have learned that in embryo sac here, that's the triploid, primary endosperm nucleus to endosperm tissue is formed, and that gives the nourishment. So this nucellar cells embryo now derives the nourishment from this endosperm. So that's why nucellar cells surrounding the embryo sac start dividing, protrude into the embryo sac and develop into embryos. Extra, this is the integument black color lining. Sometimes, not always nucellus, but sometimes that's the integument cells are also there. that integument cells divide and again protrude towards that embryo sac to derive the nourishment from the endosperm. Next one, that significance of apomixis. Significance of apomixis in that we know that to get the desired characters, the botanists have developed that hybrid seeds. But if that hybrid seeds sown into the soil and then that allow the normal gamete formation, pollination, fertilization, then in their progeny, the chances are there their segregation of characters due to segregation of chromosomes during meiosis, because meiosis is required for gamete formation. So hybrid seeds when sown into the soil, segregation of characters in the progeny. So that hybrid characters now, desired hybrid characters now do not maintain in that further progeny. So how can we maintain that? For that, this apomixis concept applied over here. If by some way that this hybrid seeds convert to apomictic, hybrid seeds convert to apomictic, then no segregation of characters, because apomixis we know that no fertilization, no meiosis. If no meiosis, no crossing over, no characters change, no variations are there. Whichever characters present in parent cell, same that's further carry forward to their progeny. So this is the significance of apomixis. For that scientists active research is going on, and scientists do what? They identify the part of the DNA which has this apomictic genes, isolated that part and then inserted into the other plants so that can also become apomictic. Next, by using this technique, farmers can use this crop year after year and no needs to purchase that hybrid seeds which are very expensive, which are available in the market. Active research, understand the genetics of apomixis. Genetics of apomixis like this, identify that gene, isolate that gene and insert into other. And to transfer the apomictic genes into hybrid varieties so that parental characters will retain into further progeny generation after generation.